to thank everyone for joining us today. I'm excited to be given the opportunity to discuss the power of light sheet microscopy in the neuroscience world and introduce you to Miltenyi's newest light sheet microscope, the Blaze. Here's an outline of what I will cover in my talk. First, I will introduce you to light sheet microscopy and how this technology can benefit your imaging workflow. I will then provide you with an introduction into the UltraMicroscope Blaze, which is our new fully automated light sheet instrument. I also want to give you a little bit of context in terms of sample preparation. So I will give an overview of tissue clearing, why and how we clear. And finally, I will touch upon some practical applications for 3D imaging in the neuroscience field. So what is light sheet microscopy? Light sheet microscopes work as virtual microtomes, almost like a tissue scanner. With the UltraMicroscope Blaze, the sample is illuminated from the side using a sheet of light. The fluorescent signal is then collected along the vertical axis, something we refer to as the 90-degree principle. For imaging, the sample is moved through the light sheets as shown in the animation on the right, resulting in a high-resolution 3D dataset. But how might this imaging technology be important or benefit you in your imaging workflow? If you are currently performing traditional wide-field microscopy, you are likely quite familiar with these tissue sectioning instruments. As you know, opaque tissues can be cryosectioned into very thin slices, something around 10 to 20 microns, which ultimately makes them translucent. And it's this translucent property that makes the sample suitable for imaging on slides. If you're currently using this strategy to image your samples, you know how tedious this work can be, and not to mention the damage the tissue can endure from the sectioning blade, as well as temperature-related issues. It is also common that during this process, sections can be lost, and therefore data is lost. It is true that 2D immunofluorescent imaging is currently the most common imaging practice. However, the data obtained represents the three-dimensional nature of the biology in a very limited manner. In comparison, renderings of a whole intact organ can provide the full picture. This can be observed here in these images. Both samples were stained for the vasculature marker CD31, but it is clear that the 2D analysis on the left cannot capture the complexity of the blood vessels in the mouse brain as one can appreciate of that on the 3D analysis on the right. Another common method for imaging larger samples is confocal microscopy, which generates optical sections instead of physical ones. Confocal microscopes do this by a point scanning mechanism across the sample. Since the entire sample in the Z dimension is illuminated, a pinhole in front of the detector is necessary to eliminate out-of-focus light. So even though you are illuminating the entire sample volume, only fluorescent signal very close to the focal plane is resolved and detected. In addition, since much of the light from the sample fluorescence is blocked at this pinhole, the increased resolution you get from the optical section is at the cost of decreased signal intensity. So to offset this drop in signal after the pinhole, the light intensity is detected by a sensitive detector, which basically serves to transform the light signal into an electrical one. So while confocal microscopy is a valuable technique for some samples and/or applications, there are a few drawbacks with regards to acquisition speed and the increased potential for photobleaching samples during an imaging session. Now to compare, with light sheet microscopy, the excitation and detection paths are uncoupled and oriented perpendicularly, where the excitation beam illuminates the sample from the side and the detection occurs on the vertical axis. In clear tissue, the excitation beam can propagate through the width of the sample, and therefore the light sheet itself is creating the optical section. This means there's no requirement for blocking out-of-focus light with a pinhole or amplification of the signal with a sensitive detector. Instead, light sheet microscopes are able to acquire the entire field of view in a single image. This gives way to much faster acquisition times, and it's a lot more gentle to your sample in terms of photobleaching. Now that we're all a little more familiar with light sheet technology, I'd like to introduce Miltenyi's UltraMicroscope light sheet platform. We have 3 different configurations, with the zoom body here on the bottom left standing as our original UM2, the Super Plan, which is the 2nd generation UM2 on the bottom right, and then we have the UltraMicroscope 3 Blaze on top. All UM systems are very easy to use and flexible to your various imaging needs. The light sheets are optimized in these systems to provide you with homogeneous illumination of your sample. This is largely due to the fact that there are a total of 6 sheets, 3 on the left and 3 on the right. They're all compatible with all clearing protocols. I will speak to this a little more in the coming slides. And they can accommodate both small samples like organoids of only a couple millimeters or larger samples like an entire mouse brain. And with the newest member of the ultramicroscope family, the Blaze, you can image samples as large as an entire adult mouse. I will now continue to focus on the Blaze, and I really want to highlight some innovative features that have been incorporated into this specific UN model. The Blaze is fully automated light sheet microscope, which eliminates several pain points that can occur during your imaging sessions with some of these features. First, the automated sample release, giving you easy and fast access to your samples with effortless sample mounting. The automated objective lens changer. The objectives live on the instrument, so there is no need for mounting and dismounting. This dramatically reduces the potential for damaging these objectives. And then we have the automated focusing feature. There are no coarse or fine adjustment knobs. Focusing in on your sample is accomplished with just a click of the button in the software. Much of this automation is conducted through the automated detection unit, which is controlled completely through the software. Inside the unit itself, there are 4 intermediate lenses providing a range of magnifications that can be applied to each objective. We have the multi-immersion plane objectives. This system is equipped with a 1.1x, a 4x, and a 12x objective. These lenses are specifically designed for light sheet microscopy, having the longest working distances relative to their numerical aperture. We have our cutting edge light sheet optics. As mentioned previously, there are a total of 6 light sheets, 2 central beams with 4 additional sheets directed at an inward angle. This is to reduce shadowing effects and ultimately resulting in homogeneous illumination of your sample. And then we have that large automated chamber, which provides plenty of space to either mount multiple smaller samples for batch imaging, as shown in the image here, or a very large sample, something like an entire human kidney. One feature I mentioned earlier is that the ultramicroscopes are compatible with all clearing protocols. But why do we clear tissues? And how is tissue clearing accomplished? So let's start with the question why. In a given sample, light passage is highly impaired due to things like reflection, refraction, scattering, and absorption, as it encounters different materials within the tissue. These materials are largely referring to water, proteins, and lipids. And each of these molecules have a different refractive index. The term refractive index, which is a term you will hear quite often with regard to tissue clearing, is a numerical value that is assigned to these different molecules and represents the speed at which light travels through a given material. For example, water has a refractive index of 1.3, proteins around 1.5, and lipids around 1.4. This number is an expression of how much slower light moves through a substance compared to that of a vacuum. The higher the number, the slower the light travels in that material. So the goal of tissue clearing is to render the tissue transparent. But what does transparency really mean? It is essentially the result of homogenizing or matching the refractive index of the entire sample. So the reason we clear tissue is to remove light scattering molecules and essentially alter the tissue composition so that the light's behavior is normalized as it travels through the sample. So if you look into literature now, you will notice there are many different clearing protocols, all a little unique in their mechanism for homogenizing the refractive index. But here I will introduce you to 3 major classes, that being the hydrophilic clearing methods, hydrogel-based clearing methods, and then hydrophobic, or also known as organic clearing methods. Hydrophilic clearing techniques start by removing lipids with some form of detergent. The samples are then placed into high concentration urea solution and imaged in an RI-matched aqueous immersion. The resulting refractive index of samples cleared in this method range from about 1.38 to 1.48. So these methods are typically quite simple and can clear tissues rather well, while also preserving endogenous fluorescence. The main disadvantage is the time it takes to clear samples, which with this protocol can take weeks. And it isn't always as effective as other methods like the organic solvents or the hydrogel-based clearing on dense samples. Hydrogel clearing methods start by cross-linking proteins to a hydrogel matrix for initial sample stabilization. Lipids are then extracted with some detergents, and it's this portion of the protocol that can take quite some time when done passively. As an alternate, and to speed up this process, active clearing can be conducted, which utilizes an electrophoresis operation. The resulting refractive index of samples cleared with this method is around 1.45. So people select this method largely because it is non-toxic, it is compatible with immunolabeling, and it also maintains that endogenous fluorescence. Some of the big disadvantages here are typically cost associated with both the imaging solutions and commercial electrophoresis systems to ultimately achieve that active clearing. Hydrophobic, or also often referred to as organic clearing methods, first dehydrate the samples using some alcohol gradient where lipids are also extracted, and they're finally cleared in a high refractive index organic solvent, around 1.55, which is also similar to that of the refractive index of proteins. The major advantages to organic clearing methods is that the resulting transparency is quite robust compared to other methods, and it is relatively easy and cost-effective workflow to adopt. They're widely used in neuroscience, really rendering those brains quite transparent. The disadvantage here is that most of these hydrophobic methods use toxic chemicals in that final clearing step, which some users like to avoid. So when it comes to tissue clearing, I hope I convey there is not a one-size-fits-all protocol. Selection of your clearing method will depend on the tissue species, your intended application, amongst other considerations. There will always be the possibility that you may have to adapt or modify your protocol to achieve true refractive index homogenized samples. Not all protocols will render samples clear enough for light sheet and therefore may not be compatible with this technology. But there are a lot of resources out there, including myself, to help you navigate and find the method most suitable for your application. Now that you have a general idea about tissue clearing, I want to shift gears a little bit to antibody selection when it comes to 3D immunofluorescence. I think it's important to point out that the time and effort to optimize traditional immunolabeling conditions for 2D imaging on slides can differ quite a bit when applied to large samples in combination with clearing. We will use this cerebral organoid stained with beta-tubulin 3 as an example. The left image was the result of immunolabeling under the specifications intended for 2D immunofluorescence. This pattern of antibody bound largely to the perimeter of the sample is a common result for many of those new to 3D imaging. From this point, you would have to optimize the immunolabeling conditions by trying to understand if this effect is due to insufficient permeabilization methodology, the wrong antibody concentration, or antibody incubation times. So though the image on the right was stained with the same beta-tubulin 3 antibody, the conditions have already been optimized for 3D intact samples, where you now observe proper diffusion of the antibody into the interior of the organoid. So if there's an option to purchase an antibody that has been previously validated for 3D immunofluorescent applications, either by another lab or a vendor, taking this route can save you both time and effort. Another consideration that can also complicate antibody optimization efforts is if you're using an indirect method for immunolabeling, with the primary antibody against your target, which is recognized by a secondary antibody conjugated to that fluorescent dye. You can see the timelines outlined here, where indirect labeling systems can increase your protocol by a week or maybe even more. So if you have the option to go with a primary antibody that has the fluorescent dye directly conjugated to it, it's in your best interest. Not only does this decrease the protocol timeline and simplifies optimization efforts because it's one antibody instead of two, but we have also seen with our direct conjugate primary antibodies that signal to background ratios are enhanced, as you can see in this comparison image on the right. To address this bottleneck in tissue clearing and 3D immunofluorescent workflows, Miltenyi Biotec has created a simple and streamlined solution to help you get up and started with light sheet imaging. First, we have our Max Tissue Clearing Kit on the left. These reagents have been developed for the hydrophobic clearing of tissues, and of important note, this kit uses non-toxic organic solvents for clearing and imaging. We are also providing an ever-growing list of antibodies that have been validated for 3D immunofluorescence. Some important highlights to Miltenyi's 3D antibody portfolio is that they are all directly conjugated to a small photostable dye, and they're recombinantly engineered, so you can feel confident that you'll get that lot-to-lot consistency between experiments. Our research and development department has worked really hard to develop a full solution to your 3D imaging needs. This includes predefined protocols that are supplied on our webpage for free for the optimal immunostaining and clearing of different tissue species. Here's just an example of a workflow for one of our protocols that will walk you step by step through the immunolabeling portion and the clearing portion of intact mouse brains. We are now moving into the last portion of my talk where I will review some neuroscience-based applications using the ultramicroscope, but of course there's potential for much more than what I will show you here. I hope that you feel free to reach out to me. I'll provide my contact information at the end of this presentation, or you can join us at the booth afterwards if you want to discuss ways of enhancing your research projects by incorporating light sheet microscopy. So I'll first hit on some of the most popular areas of neuroscience research where light sheet has proven to really be a powerful tool. Um, in Alzheimer's and Parkinson's disease research, the 3D imaging of intact mouse brains is allowing researchers to quantify plaques and dopaminergic neurons with the goal for identifying target sites and therapy approaches. The brain that you are looking at now is that's stained for Th positive cells, so those dopaminergic neurons. So in stroke research, these 3D renderings are quite powerful tool for evaluation of stroke affected regions in the brain and helping to assess therapy models of efficacy. The brain that you are looking at here, again, it's an intact mouse brain that was stained for CD31, that endothelial marker, really nicely showing that vasculature. In an attempt to understand neuronal regeneration and plasticity, these 3D images acquired from Lightsheet allow researchers to quantify neuronal plasticity using brain organoids like this one shown here. Moving up in sample size, the Blaze and its large sample chamber can accommodate 3D imaging of an entire adult mouse, shown here, which provides a really nice solution for conducting complex connectivity mapping and analysis of neuronal regeneration of an entire organism. This mouse here was a Thy1 GFP mouse. It was labeled for those GFP positive neuronal structures that you see in green. The bones and internal organs are prominent with the PI labeling in white and the muscles visualized by autofluorescence background in red. Another widely used application in neuroscience in combination with light sheet microscopy is whole brain activity mapping, which is accomplished by automated 3D image analysis and Allen Brain Atlas registration to essentially map those active neurons in an intact mouse brain. The most commonly used immediate early gene marker, CFOS, which is used in this study here, where the researchers wanted to look for brain regions differentially regulated when mice explore a new environment with or without their whiskers. But in general, this pipeline for whole brain activity mapping has countless possibilities in many different research areas such as addiction, stress, trauma, obesity. The list can go on and on. And as you can imagine, our neuroscience-based reference list is ever growing. If you are interested in reviewing some of these publications utilizing the ultramicroscope, you can find this document on our webpage. And with that, I appreciate your attention. If you have any questions, I hope you contact me. My email is listed here. You can also join us in the booth for immediate inquiries right after this. Thank you.